Hello everyone. Okay, good afternoon Sushma, good afternoon. Okay, good afternoon everyone. So friends, we are doing the enzyme and its kinetics in just two classes. Okay, in this particular class, I will do the all the characteristic features as well as the properties of enzymes, and most importantly for the exam point of view, factors affecting the enzymes. Good afternoon, good afternoon. Factors affecting the enzyme. And friends, in the tomorrow class we will be discussing the enzyme kinetics. We will be discussing enzyme kinetics in the tomorrow class. Good afternoon all, good afternoon. Okay, so as I am saying friends, this enzymes is a little bit a difficult topic, so we will do it in two classes, but I will do it faster. So in today's class we will discuss the properties and factors affecting, and followed by the enzyme kinetics. Okay, and friends, what kind of questions that we can expect in the enzymes? You can expect the part B question, you can expect the part C question. But friends, in most of the cases we can expect the question from the enzymes and its kinetics, both in the part B also friends, as well as in the part C also. Okay, so my sincere request to all of you, when you are revising the enzymes or when you are reading the enzymes, keep it in your mind. Please try to read A to Z. Do not miss any concept at all in case of the enzymes. Okay, good afternoon, everyone focus. Okay, so in this class we will completely read about the properties. Okay, whatever the points that I will highlight, I want you to remember, because friends, many times in case of a part C questions, good afternoon everyone, many times in case of a part C questions, we got the questions exclusively from the properties only. That is the reason friends, it is very important and it is mandatory that I want every student has to remember the properties without fail. So please focus on that. Okay, now listen to me carefully. What are enzymes? Friends, I will go little bit faster, not too much fast. I'll go little bit faster, by that we can finish it little faster. We know very well friends, the moment you heard the name of a enzyme, only 2 points has to come in your mind. Enzymes are the biocatalyst. Ultimate goal of the enzyme is what? Good afternoon, good afternoon. Ultimate goal of the enzyme is increasing the rate of reaction. We all people know very well how does this particular enzymes are basically increasing the rate of reaction. They are increasing the rate of reaction friends by lowering the energy of the activation. In a simple words, enzymes are specific in nature, which means what? Every student remember, one enzyme function cannot be replaced by the another enzyme. Yes or no friends? Suppose example, phosphofructokinase enzyme function cannot be replaced by the pyruvate kinase. Similarly friends, pyruvate kinase enzyme function cannot be replaced by the phosphofructokinase. Is it or not? Of course yes. Now, enzymes are the proteins that increase the rate of reaction by lowering the energy of activation. We all people know very well, and every student has to remember that. We know, right friends, we are having 37.2 trillion cells in our body. Friends, if this particular 37.2 trillion cells, if they want to do the metabolic reactions, if they want to perform any kind of a chemical reactions, compulsory what will be requiring friends? We will be requiring the enzymes. So what is the statement I would like to conclude? Without enzymes, not even a single chemical reaction or not even a single metabolic reaction that is taking place inside the body. Now whenever a reaction is occurring, suppose example, I want every student has to listen to me. An example, whenever a substrate is converting into product, every student listen. Do you think substrate can change the concentration of enzyme? Do you think product can change the concentration of an enzyme? Not at all. So what is the statement you have to remember? The enzymes are not altered or consumed during the reaction. That is one of the major characteristic feature of a enzyme. And they can ask a simple statement that, friends, all the enzymes are reusable in nature. What is the statement? All the enzymes are reusable in nature. Undoubtedly, my friends, which is absolutely true statement only. Okay, now friends, now you can ask me one valid question. Everyone, please listen my statement carefully. You all has to get one question. Sir, does every chemical reaction can occur without enzymes or not? Once again, listen. Does every chemical reaction will occur without enzymes or not? I can give the answer yes. Every chemical reaction can occur without enzymes, but friends, that reaction will take a greater time. Suppose example, everyone listen. If a particular reaction wants to occur without enzyme, it is taking half day. Without enzyme, a particular reaction wants to occur, it will take half day. Whereas if there is a presence of enzyme, it will happen in 1 second. Presence of enzyme will make that particular reaction to occur in 1 second. So what's the statement you have to remember? Enzymes are the one which are making the reactions to go faster. So which doesn't mean that without enzymes reactions will take place. With enzymes reactions will occur very faster. And friends, every student remember, enzymes are present in the biological system. Now we all people know that the biological systems are very sensitive to the temperature also, and at the same time, friends, the biological systems are very sensitive to the pH also. We all people know that. So what is the statement we can tell? We can give a statement that enzymes are highly sensitive towards the temperature also, as well as towards the pH also. Now that's why especially our enzymes will play a key role in the metabolism, diagnosis, even therapeutics. Let's take an example of a diagnostic tool. Let's take an example of a ELISA technique. Let's take an example of a RIA technique. So in the ELISA and RIA, everything is what? Enzyme dependent reactions only. Correct na? Take an example, friends. Take a best example of a myocardial infarction. See friends, whether the particular person having the myocardial infarction or not, we can easily confirm by the presence of the level of the enzyme. What, my friends? By the presence of the enzyme level, we can easily tell whether that particular person having any chances of getting a cardiac arrest or myocardial infarction condition. So what I mean to say here, the enzymes plays a key role even in the diagnostic also. That's what I want you people to remember. And friends, from where we can expect the part B questions? From here. From here only we can expect the questions, friends. That is the features, what are the characteristics. Now you only listen. Common features for enzymes and inorganic catalysts. That is the major difference between the inorganic catalyst and enzymes, is that inorganic catalyst also will allow the reaction to occur, but inorganic catalyst will allow the reaction to occur very slowly, whereas friends, enzymes allow the reaction to occur at a very faster rate. That's why every reaction in our body, they won't prefer all the time inorganic catalyst to allow that reaction. Instead they will always prefer enzymes to carry that particular reaction. And what did they tell to all of you? They are not used. At the same time, they are not changed during the reaction. Don't get confused, my friends. And please listen, they don't change the position of a equilibrium. This is most important. And the direction of the reaction. This is, among all, this is the significant point. Which means that enzyme always allow the reaction to occur only in one direction. And second point, enzyme never disturb the equilibrium. If the reactants are more, enzymes will be helpful in balancing the product. If the products are more, enzymes will be helpful in balancing the reactants, which means that it will always try to allow the equilibrium of that particular reaction. Please don't get confused with that particular point. Friends, we all people know, right? If the enzyme wants to act, the enzyme has to combine with this substrate molecule. at which condition or the what state the enzyme can combine it with the substrate complex, which we can call, friends, a transient complex. All the students, please listen my statements without confusion. Enzyme, when it is associated with the substrate, the state at which the enzyme associating with the substrate, we can call it as a transient complex or the transition state. And most of the students will think that enzyme and substrate will attach us very tightly, which is absolutely wrong statement. You know, enzyme and substrate, they are not associated very greatly with each other. What is the reason, friends? The reason is all the interactions are the weaker interactions. Because of the weaker interaction between the enzyme and substrate, their attachment cannot be stronger. Initially the interactions are weaker, later at the stabilizer condition the interactions will become the stronger. Now friends, listen. Suppose example, friends, this is an enzyme. Okay, this is an enzyme. Do you think the substrate will come and associated to the every part of the enzyme? That is not at all possible. If a substrate wants to attach us to an enzyme, it should have a particular site, the particular part or the particular site at which enzyme will come and attach us. That site only we can call it as the active site. Otherwise we can call it as a pocket like structure. Otherwise we can call it as a cleft like structure. And most of the students will ask me, sir, does enzymes are larger as well as the proteins are larger? See, we all people know that enzymes are proteinaceous in nature only, but what is the statement I can give you that enzymes are the very larger proteins. Now why I am highlighting this point, if the enzyme is very larger, so probably the active site also should be very larger. No friends, only the enzymes will be larger, friends, but not the active sites. Active sites are very small sites there. Even though enzyme bigger, active site is extremely smaller. Because friends, you have to listen to me. What is the active site contains? Active site contains the place of or space of 8 to 10 amino acids. That's all. Whatever the substrate that is attaching, my friends, that substrate should not contain more than 8 to 10 amino acids in size. If it is more than that, it cannot fit into that particular active site. So remember that even though enzymes are very large protein, my friends, undoubtedly the active site will be very smaller. Do not get confused with that particular point at all. Now all the students listen to me very carefully. This is an enzyme, this is the substrate. Substrate exactly fit into the active site. All the students listen. Whenever the substrate come and fit into the active site of the enzyme, what we can call it as? We can call it as the ES complex, enzyme substrate complex. Now you can ask me a valid question, sir, how does the substrate can be converted into product? Every student remember, the reaction is very simple. What is that? Only two things, either making the bond or breaking the bond. That is only function of an enzyme, either making the bond or breaking the bond. Suppose you tell me, if I want to form the sucrose, what I will do? I will add the glucose and fructose, then I will get the sucrose. So what I am doing here? I am making a bond, a glycosidic bond. Now I want to do the opposite. I want to break down the sucrose into glucose and fructose. Then what I will do? I will do the breaking of the bond. But what is the point every student has to remember? Either making the bond, my friends, or breaking the bond, my friends. Everything is under the control of the enzyme only. So in one point every student remember, enzyme can convert the substrate into product either by making or breaking the bond. Very simple point it is. Now where a particular substrate will come and attaches, friends, we discussed that the substrate will come and attaches to the active site. Now friends, it is very important for the part B. You have to remember this without fail. What are the characteristic of an active site? Please listen to me, friends. Absolute see, characteristics means how a particular enzyme or how a particular substrate attaching to the active site of an enzyme. That is what we are explaining. So there will be an absolute specificity, there will be a group specificity, there will be a stereo specificity. I will explain everything elaboratively, but you have to remember 3 compulsory. Just now I have explained you, right? Friends, enzyme initially attaches to the substrate at the active site of the substrate. See, look into the picture. This is the active site. To the active site of the enzyme, substrate can bind, look into this, by the multiple weak interaction. Most of the students will think that the attachment is a stronger attachment. Then they will think that the attachment will be a covalent bond. No, no, no. So always keep it in your mind. That is what we generally call it is the non-covalent bond. Do not get confused. Now I am going to explain this particular slide. I am requesting all of you friends, remembering this particular slide is important for the part B. What we discussed, there are 3 types of specificity: absolute specificity, group or relative specificity, and friends, linkage specificity. It is very easy. Now listen, absolute, you have to remember, absolute means the enzyme can catalyze one type of reaction for a single substrate. What do you mean by that? All the students look into the diagram. What is this only single substrate? You all people know very well, friends. What is the function of a urease? Urease is the only one which can break down the urea. We know that. Now you tell me, does the urease can break down the ammonia? That's not possible. Do you think urease can break down the uric acid? Even that is also not possible. So what's the statement you have to remember? Urease is catalyzing only one single substrate. What is that substrate? That is the urea. That's why we will be calling this as the absolute specificity. It is the one which is catalyzing one type of reaction for a single substrate. Do not get confused with this particular point at all. Now the second one, group specificity. What do you mean by the word group specificity? It is catalyzing one type of reaction for a similar substrate. Once again listen. Same friends, there is no difference, but what it is doing, a group of similar substrate. Now let me give you one wonderful example. Let's take an example of a hexokinase. Now you tell me, what is the function of a hexokinase enzyme? Kinase is the enzyme which is adding the phosphate group, of course, but we have used a word hexokinase. So friends, the hexokinase enzyme is going to add the phosphate group only to the hexoses. That's the unique statement. Now, do you think the hexokinase enzyme will it add the phosphate to the pentoses? No, it never. Hexokinase enzyme is very specific to, see, the function is adding phosphate, no doubt about it. But it never add the phosphate group randomly to everything. It will add the phosphate only to the hexoses. Correct, na? So that's what we can call group specificity. Friends, don't get confused. It's very important you have to remember the examples. Linkage. Friends, the moment you heard the name of a linkage, what is the point has to come in your mind is, linkage is basically representing the bond. Okay, now you only tell me, understand what I am talking. Chymotrypsin. What's the function of a chymotrypsin? Chymotrypsin is break down the peptide bond. We know that very well. Okay. Now, an example. Let's consider ester bond. Ester bond. Let's consider glycosidic bond. So friends, ester bond can be cleaved by what enzymes? Lipases. Glycosidic bond can be cleaved by which enzymes? Glycosidases. So what is the statement you people has to remember? Do you think, friends, glycosidase enzyme, do you think it can cut the peptide bond? That is not possible. Do you think glycosidase bond can cut the ester bond? That is not possible. Do you think glycosidase can cut other bond except glycosidic? No. It is specific for only glycosidic bond. Similarly, friends, chymotrypsin specific for peptide bond, lipases are specific for the ester bond. This kind of specificity only, friends, we will be calling it as the linkage specificity. The moment you heard the name of a linkage, remember it is representing the bond. and group means group of substrate, absolute means single substrate. Remembering examples is important for the exam. Okay, now friends, focus, let me go little faster. Substrate. Now you can ask me a valid question, sir, what is the difference? Video is not clear. Okay, I think video is clear only, Apurva. I want you to refresh. For everyone video is clear. It is blurred means maybe your internet connection. Video is very clear, Apurva. Please just refresh and rejoin. See friends, what is the difference between the substrate and reactant? According to me, video is very clear. Reactant. What's the difference between the substrate and reactant? Friends, both are same, but whenever a substrate, if it is involved in the biochemical reaction. What, my friends? If a substrate is involved in the biochemical reaction, then only we will be calling this as the reactant. Please kindly don't get confused between the substrate as well as the reactant. Okay, please continue. Now you only look into the picture, friends. Substrate will go and fit into the small active site of the enzyme. Now all the students listen, let me explain you very fast. To explain what to explain how the substrate can fit into the active site of the enzyme, there are many models were proposed. My friends, among all those models, there are two models are most important models. One we can call, friends, lock and key model. Another one we can call, friends, induced fit model. Okay, don't get confused. Now we all people know that lock and key model is the old model and it is not acceptable for the all enzymes, especially regulatory enzymes cannot follow the lock and key model at all. Whereas induced fit model will be the new model which is applicable to the all enzymes. Anyway friends, the name itself very clearly indicating what is lock and key model. Do not get confused with this particular statement. You all people know, right? Lock and key. If I want to open a specific lock, I can't keep a random key. We all people know that. Whatever the key that fit into that particular lock, I can open that lock that particular with that particular key only. Same way, every substrate cannot fit into that active site. That's not possible. So suppose, because what is the reason? The reason is very simple. The active site is the rigid in nature. This is the point every student has to remember. What, friends? The active site is the rigid in nature. Because of the active site which is rigid, whatever the substrate that exactly fit into that particular active site, friends, so only that particular substrate can fit over there. That substrate only converted into product. But do you think every substrate is designed in such a way that can it fit all the time into the active site? That is not even possible. That's why, friends, this lock and key model does not applicable for any kind of the enzymes. 90% of the enzymes, they won't follow this. But why most of the enzymes will follow only this induced fit model? The reason is a very clear-cut reason, because it is flexible. Friends, when I use a word flexibility, what you have to remember, flexibility means according to our convenience we can convert it. See here, all the students look into the picture. This is how the active site is looking like. But at the same time you can also see, my friends, how does this particular substrate is looking like. This is how an active site is looking like. This is how a substrate is looking like. According to the lock and key model, do you think this substrate can go and fit into this particular active site? No, that is not even possible. But what I want you people to remember, this particular substrate, all the students listen, this particular substrate can come and attaches to this active site. Then what is going to happen? Since the active site is super flexible, the active site can be converted according to the size of this particular substrate. Good afternoon, Deepika, continue. So that's what we can call it as what, friends? Flexible model. So according to the induced fit model, every student listen, according to the induced fit model, it doesn't matter whatever the structure of a substrate is, whatever the shape of the substrate is, doesn't matter. Once the substrate just binds to the active site is changing its shape in such a way that substrate can easily fit into that particular active site. Kindly do not get confused with that point, which we can call induced fit model. Don't confuse at all. Okay, now friends, all the students listen to me very carefully. Enzyme catalyzed reactions, we all people commonly know that enzyme combined with the substrate. What do we call this as? Enzyme-substrate complex. My friends, this is what I am saying here. This is only I want you to remember. Here the type of interactions are, my friends, undoubtedly multiple weak interactions only. They are not at all the stronger interactions. Here, now this is the point every student has to remember. The transition state. You please imagine what I am talking, friends. The transition state, what is happening? Either making of the bond is happening, or friends, breaking of the bond is happening. Either making of the bond or breaking of the bond is happening. Listen to me, friends. Whenever there is a making or breaking is happening, now you people has to remember, in this transition state, the enzyme plus substrate attachment will be the covalent. Initially when the enzyme just attaches to the substrate, here it is the weak interaction. But in the transition state where the making or breaking is occurring, that time the interactions are very covalent. Then it's a kind of stronger interaction. So because we have to break the strong bond only, or we have to make the strong bond only, then only, friends, we can get the product. Don't get confused with that particular statements. Friends, once we form the product, what is going to happen? The product is getting dissociated. Friends, once the product gets dissociated, once the product is formed, focus. Once the product is formed, do you think product can attach us to the active site of the enzyme once again? No, that is not at all possible. Please keep it in your mind. Because the product do not have the affinity, the product do not have the binding site with the enzyme. So the product is released and now enzyme is getting ready. For what purpose it is getting ready? For the substrate. So what's the point you have to remember? Like this, in 1 minute, I am using the word only 1 minute, our enzyme can do the millions of reaction. Even we can say that, friends, more than a million. So in 1 minute I am talking about. In 1 minute it will perform millions of reactions. That's why we can use a word. Enzymes are the fastest molecules in the cell, which make the reaction to occur very fast at high accuracy by reducing the activation energy. We will discuss what is activation energy. And friends, from where we are getting the part B question, okay, and part C question also. You can expect the part B also, friends. Even you can expect the part C also here. I don't know what you do, my friends. It's my sincere request. I want you people to remember this without fail. So just pay attention. Okay, friends, please listen. Whenever you heard the name of a holo means what? Complete enzyme. Correct, na? Holo means complete enzyme. Don't get confused, friends. Don't get confused at all. So when an enzyme is said to be a complete enzyme, friends, please listen. Please listen every statement. From 11th standard, we are reading a statement that enzymes are protein in nature. What is the statement we are studying? Enzymes are protein in nature. My friends, there is no doubt, 100% true. Enzymes are protein in nature, no doubt about it. But I never use a word. Enzymes are made up of 100% of protein. That is wrong statement. What is the statement I can give you? I can use the statement that 80 to 90% of the enzyme is the protein, plus 10 to 20% of the enzyme is the non-protein part. Now every student has to remember a unique statement. You can ask me a valid question. Sir, 80 to 90% of the protein is made up of enzyme only. Do you think without the non-protein part, does the enzyme become active? I can give the statement absolutely not. Even though it is 10 to 20% of the non-protein part, that non-protein part only most important. What will be the reason? The reason is a very simple, because the non-protein part only will form the active site. This is the major reason. Which part, friends? Non-protein part only making the active site. That's the reason only we can call it 10 to 20% of the non-protein part only most important part. And EPO enzyme, you please remember without fail, which we can call protein portion. Here we have written very clearly. What we have written very clearly, EPO enzyme, which is a protein portion, absolutely inactive. Such a nice statement it is. Now cofactor, what we can call the non-protein portion. Don't get confused with this. Now when the EPO protein combined with the cofactor, which is the non-protein part, then only we can call the holo enzyme. So when we can say an enzyme is an active enzyme, we can say an enzyme is an active enzyme, friends, when it is complete enzyme, which we can call the whole enzyme. All the students, please look into this and try to remember. Here you no need to use your brain at all. Here you have to remember. The enzyme contain a protein part called as EPO enzyme. Enzyme contain a non-protein part, we can call it as the cofactor. Cofactor, which is the non-protein part, of two types. This is also important. You people has to remember it compulsory. The cofactor or a non-protein divided into coenzyme and divided into prosthetic group. Divided into coenzyme as well as the prosthetic group. What's the difference? You have to remember. The moment you have you heard the name of a coenzyme, keep it in your mind. It is a organic. Always remember, always. Organic molecules are very large. Yes or no? If large organic molecule, undoubtedly, friends, their attachment is cannot be tighter attachment because very large. So their attachment will be the loose. Friends, when you look into the prosthetic group, what is the point you have to remember? They are small inorganic. Okay, but since they are very small, right, my friends, then their attachment is absolutely tight attachment. This is the statements I want you people to remember without fail. And for the exam point of view, you have to remember the examples that what we are going to discuss now. All the possible examples. What are the type of cofactor? Coenzyme and prosthetic group. Both are non-protein only. One can attach us loosely, one can attach us tightly. Now friends, if I ask you to give me the examples of the coenzymes, which is the non-protein part, please remember most of the vitamins. Most of the vitamins, because we read, right, if an enzyme wants to work, there will be a requirement of the vitamin. Why? Because that vitamin only acting as the non-protein part, that vitamin only required in the active site. Don't get confused with that particular point. Now I don't know what you do. It is your responsibility, compulsory, mandatorily, you have to by heart this particular slide. We have already solved a question. I am not solving one more time, which is asked for 4 marker and also asked for the 2 marker. I am repeatedly requesting all of you, don't skip it. I mean to say, don't neglect it. Read it without fail. Read 2 times, 3 times, 4 times until you remember it without fail. If you can't, actually you have to read all. But for your better, for the better revision, I am highlighting 5. At least read that 5 without fail. Cytochrome oxidase, which is electron acceptor 4, which is present in the mitochondrial membrane. We have seen a particular question in the biochemistry in the morning class. They have asked, right, Which of the following do not have the iron and sulfur as the cofactor? I hope whether you remember or not. I hope you all remember. Morning only we seen a question in the biochemistry only. Iron and sulfur are not present where? In the cytochrome oxidase. This is the exact question they have asked in the 2022 February paper. Cytochrome oxidase do not have the Cu2+. There is one more type of cytochrome oxidase is there. This one. This cytochrome oxidase, it contains the Fe2+ or Fe3+. But this cytochrome oxidase is not present in every single organism. So please don't confuse, friends. If they ask you a question, Fe2+ or Fe3+ are utilized by the cytochrome oxidase or not, you can give me the answer that of course no. 90% cytochrome oxidase only uses the copper. If not copper, they will use the heme. In very rare cases, it will use the Fe2+ or Fe3. But catalase, peroxidase, they will compulsory use the Fe2+ or Fe3+ as their major cofactor. Cytochrome oxidase majorly prefers either copper or heme. Pyruvate kinase, which is involved in the most important step in the glycolysis, that is conversion of phosphoenol pyruvate to pyruvate, very important step, and it will be requiring potassium as the cofactor. Most important enzyme, the first enzyme of the glycolysis, hexokinase, glucose 6-phosphatase, pyruvate kinase, it will also use the magnesium. So here pyruvate kinase is using the magnesium also, pyruvate kinase is using the potassium also. You have to answer the question according to the options that they have given in the exam. Okay, remember these 4 without fail, friends. And the 5th one, dinitrogenase enzyme, which we have studied in the nitrogen metabolism, correct now, which is the molybdenum. Don't confuse, friends. Urease will utilize the nickel. Carbonic anhydrase and alcohol dehydrogenase will use the zinc. Friends, I am requesting all of you 1, 2, 3, 4, 5, 6, 7. At least remember this 7 enzymes and what are the cofactors that they are using without failure. Nothing to explain. I want you to remember that. So now let's talk about the different cofactors. We discussed that cofactors are of 2 types. One type of cofactors we can call coenzymes. Another type we can call it as the prosthetic groups. First we will discuss about the coenzymes briefly. Then we will talk about the prosthetic groups. Friends, the moment you heard the name of a coenzymes, what's the common statement in the exam also? By looking at that only you can answer the question. Coenzymes are the one which is actually acting as a group transfer agents. Very simple, right? So what they are transferring? They can transfer hydrogen, electrons, not only a single atom. They can transfer even a group of atoms also. You have to remember these coenzymes further categorized into metabolite coenzymes. They are further categorized into, friends, vitamin derived coenzymes. Don't get confused with this particular point at all. Now friends, we all people know that we animals or we mammals, we can't synthesize the enzymes. But you have to remember the importance of an enzyme, why we have to take externally. Because friends, whatever the food that we are taking which contain vitamins, you know very well the vitamins we will be requiring in a very low concentration only. But what's the greatest point every student has to remember? Even though we are taking the vitamins in a very low concentration, they are having a great significance. Means they will be required for the function of many enzymes which are involved in our biochemical reactions. That's why vitamins, even though they are required in the low concentration, deficiency of that low concentration will cause many diseases. Never forget that point. Now friends, all the students pay attention. Examples of metabolite coenzyme. This is what you have to remember. The first example, I am requesting all of you remember every example. In a given part B question or part C question, you can get the questions from the examples only. The first example is the ATP. Second example is the Yedomet. Requesting all of you to remember that. Okay. So the third example is 5, 6, 7, 8 tetrahydrobiopterin. I want you to remember them. And you can easily remember that The moment you heard the name of a ATP having only one significant function, that is donating the phosphate group. Because we know very well phosphate means energy. Many biochemical reactions that are happening inside our cell, they will be requiring the energy, no doubt about it. And ADOMET, adenosyl methionine, we have studied in the molecular biology RNA processing. So what's the function of adenosyl methionine? Just it will donate the methyl group. We all people know very well methyl group is very important. It is protecting the mRNA from the degradation. Upon donating the methyl group, this is also we have studied, adenosyl methionine can be converted into homocysteine. I am teaching little faster because you already read it. But my request is to remember 5, 6, 7, 8 tetrahydrobiopterin can act as an important enzyme for the nitric oxide synthase. What is the nitric oxide synthase function? We all people know very well, when an amino acid arginine can be converted into citrulline, what will be released as a byproduct? The NO can be released as a byproduct, correct na? And what will be the enzyme that is involved in the synthesis of this nitric oxide? This NO, nitric oxide synthase enzyme. If this nitric oxide synthase enzyme wants to be active, they will be requiring 5, 6, 7, 8 tetrahydrobiopterin as their major cofactor, because this only we can expect in the matching questions. Okay, now friends, let's talk about the vitamin derived coenzymes. You can tell me very easily now vitamin derived coenzymes. You can take an example of most important ones. NAD you can take, as well as the FAD you can take. What is the point you have to remember? Examples of that particular enzymes. That only you remember. No need to put extra effort here. Nicotinic acid or niacin. Please remember that. Only that you remember, friends. Required or acting as a precursor for the synthesis of NAD and NADP+. First point. Second point, every student has to remember whatever the 2 points that I am highlighting. I am requesting all of you to remember the 2 points. NAD and NADP are acting as the coenzymes for the dehydrogenases. Friends, these chapters are rigid chapters. 90% of the students trust my words. Blindly trust me. The people who think we are overconfident, we can remember everything, they can answer very conceptual questions very easily. Trust my words, they will blindly make the mistake in this type of questions, because these are all rigid concepts, so they don't show interest and they never read. So if you want to score, you have to remember these kind of statements, only very important in our exam. So remember, NAD and NADP are coenzymes for this particular dehydrogenase enzyme. Don't get confused with the point. Now friends, FAD and FMN. Please remember. FAD and FMN. Please remember. Flavin adenine dinucleotide, flavin mononucleotide. Both can act like what? Both can act like a reducing equivalence. We know that. We also know that, friends, both can act like a electron carriers. That also we know very well. Now what's the significant point every student has to remember is that flavin, they see, please remember here I have very clearly mentioned a point that NAD and NADP plus are required for the mechanism of dehydrogenase enzymes. What is the dehydrogenase enzyme? Which is involved in removing the hydrogen. I have told to all of you many times in the glycolysis also, as well as in the Krebs cycle also. Friends, wherever you heard the name of a dehydrogenase, keep it in your mind that it is involved in the production of the NADH. We all people know that very well. Now listen to me. The moment you heard the name of a FAD and FMN, just keep it in your mind, friends. They will be involved in the oxidation reduction reactions. Okay, in other words, FAD and FMN is important for the mechanism of oxidoreductase enzymes. If this oxidoreductase enzymes wants to work, obviously what they require, friends, they will be requiring the FAD and FMN. Don't get confused with that. So first example of vitamin derived coenzyme is NAD, FAD, FMN, and the third example is the TPP, thiamine pyrophosphate. What you have to remember once again, enzyme. NAD derived from what? NAD derived from niacin. FAD derived from riboflavin. And from where the TPP is derived from? TPP is derived from the vitamin B1, which is the thiamine. Once again, listen. NAD is required for dehydrogenase enzymes. FAD and FMN is required for oxidoreductase enzymes. Correct na? Now please listen my statements. You please remember without fail. Don't worry about the structures. The TPP majorly involved in the oxidative decarboxylation. Then friends, it is also involved in the transketolase enzyme reactions. These 2 points I want every student has to remember. It is involved in these 2 reactions. Do not get confused with this particular point at all. Okay, then the last and final one, PLP, pyridoxal phosphate. I don't know what you do. It is your responsibility to remember. And majorly remember a point that pyridoxal phosphate is majorly required for the isomerization, decarboxylation as well as the transamination. Friends, these all questions are extremely important for the matching. Please don't leave that since they are rigid concepts. Okay, and once again the asked part C question. The asked part C question in the exam from this particular specific table. I don't know what you do. It's your extreme responsibility to remember this. Please don't neglect. I will at least highlight, focus on that. Coenzyme A involved in the transfer of acyl group. Please remember, lipoate involved in the electron and acyl groups. NAD hydride ion, pyridoxal phosphate amino group, TPP aldehyde. So 1, 2, 3, 4, 5. At least remember these 5 without fail for the exam point of view. I am requesting you, don't neglect. Here nothing to explain there. Okay. So friends, so far till last slide, whatever we have studied is complete structural properties of enzymes. What we have studied, friends, we have studied completely structural properties of enzyme. Now what we are going to study? Now we are going to study completely about the biochemical nature as well as the activity of enzymes. Please don't get confused with this particular point. What is the function of enzyme? We all people know that enzymes are the one which are involved in the chemical reaction. Correct na? So we all people know that enzyme actually work very effectively. Why every reaction will prefer the enzyme is because enzymes will reduce the energy of activation. Enzymes will reduce activation energy. Anything is same. First of all, how can I define the word? How can I define the word activation energy or energy of activation? Please don't get confused with this particular point. Listen, activation energy means, friends, please listen. Any reaction wants to start. Any reaction in our body, any reaction wants to occur. Before starting the reaction, we require little amount of the energy. What do we call it, that particular energy? That energy only, friends, we will be calling it as the activation energy or energy of activation. Any chemical reaction that is taking place inside the cell, any chemical reaction that is taking place inside the body requires some amount of energy to get it started. That energy called as activation energy. Everyone, please look into the diagram very carefully. This is the lactose. We know, right? Lactose is a disaccharide, so we can't use the lactose directly. So what we are going to do? We are breaking the lactose into glucose and galactose. Very simple point it is. Glucose as well as the galactose. Don't get confused. Now you only tell me, if I want to break down the lactose into glucose and galactose, what should I do? I have to break down the glycosidic bond. I have to break down the glycosidic linkage. We all people know that. Now imagine there will be a no enzyme. You try to understand. In order to break down this particular linkage, see friends how much energy that we are doing, spending. Listen my words. Of course, listen friends. Of course, without an enzyme, the reaction is taking place. No doubt about it. Without enzyme, reaction is taking place. But what is the point every student has to remember? Reaction will occur slower. Very slower. And it will take a long time. Reaction will occur slowly and it takes long time. So that much slowly, none of the reaction should occur. Then we can't perform any function in our body. But when we use the enzyme, there are two great advantages are there. What are the two great advantages? See, all the students listen. We can run the reaction faster. We can run the reaction faster and we will spend a very low energy. So we are spending energy also low and we are allowing the reaction to occur super fast. That is the reason we will be calling this as, we will be calling enzymes or the biocatalyst, which make the reaction to occur very faster by reducing the activation energy. So ultimate function of enzyme is reducing the energy. Which energy? Activation energy. What is activation energy? The energy which is required to start the reaction. By spending less energy, we can allow the reaction to occur at a greater speed. Do not get confused with that. And friends, what factors are going to affect the enzyme activity? Environmental conditions. Right now we studied, right, cofactors and coenzymes. These also plays a crucial role in enzyme activity. That's what I told you. If you don't take a particular vitamin from the nutrition, from the food, deficiency of that vitamin will never allow you the working of an enzyme. If there is no enzyme working, no biochemical reaction, which leads to very serious condition, very serious diseases. And undoubtedly, the enzyme inhibitor, whether it may be an antibiotic or it may be another drug, right? So these three affect the enzymatic activity. From where we can expect the questions. Here you can expect the part B also, part C also. We can get the, actually friends, we'll get the questions from three parts. But mostly we can expect the questions from the inhibitors. How an inhibitor is affecting the enzymatic activity. Okay. So friends, that's all for now. What we do is, in the tomorrow class, I will finish this. Okay. What I will do, in the tomorrow 11 AM class, I will finish the leftover part B questions in the 2022 paper. In the 3 PM, I will finish this and I will discuss till the Michael-Menten kinetics. And we will solve some numericals. Okay. And friends, I am requesting all of you, please take the mock test without fail. Please attempt the mock test. Please consider this mock test is a CSIR exam which you are going to give on 17th. And please participate in the mock test. Try to answer 15 questions in the part A and at least 30 questions in the part C. At least part B, 20 questions in the part C. Please answer, okay? Please take the mock test without fail. One mock test is uploaded. Tomorrow another mock test is also uploaded. Like that I will upload 6 high quality mock tests. I am requesting all of you to take the mock test. Okay friends, I will see you in the tomorrow sharp 11 AM. Okay. Thank you all. Thank you so much. Revise well, read well, at least spend 8 to 9 hours minimum on your preparation. Okay.